Okay, so next we're going to have a slight change in order. We're going to have Bjorn from the CRG, who's going to talk about functional data extrapolation across species.
Hello, I'm Bjorn Langer. I work in a group of Cedric Notre Dame and at the CRG, where we have been part of work package 3 of the BOFREC. We have been involved in task 3.5 on the comparative analysis and also in task 3.2, the standardization of data analysis pipelines. This is work that Jose then will present in 15 minutes. And I'm teasing it though here because this standardization of data pipelines is ensuring data comparability, which is the grounding and enabling comparative analysis. So what I'm now talking about is an aspect of the comparative analysis, the cow evolutionary annotation. You are aware that few species, namely human and mouse, are extensively functional annotated, but not so many others. And from an evolutionary point of view, we are wondering, can we use human and mouse to learn something about other species, particularly the cow, by extrapolating basically functional annotations? For example, by learning underlying grammar. And when I use these words like grammar and learning, many will think of machine learning, which in the recent years has been experienced a boost due to the advent of large training data sets. And with this came some interesting tools also, like Informer is on the data that has been trained on, namely human and mouse, being able to predict from an input DNA sequence a whole array of functional annotations. Another work is, for example, has been trained on human, on the categorization of ataxic regions, and be applied across species. We also have in Cedric Notre Dame's group currently some work going on about this topic of data extrapolation via machine learning approaches. But initially we were focusing on creating a baseline for approaches like this by employing established methods. For example, these two that you see here, which are part of the UCSC genome browser package utilities. So we use these sequence alignment-based approaches to project data from human onto the cow and verify this with existing cattle data. Particularly here, the FANG data that Gabrielle in the morning session already had introduced, which provides us with cattle data with various functional annotation tracks, of which we chose ataxic as an example. And this data further provides us with multiple tissues, 2 samples per tissue, of which we took the intersection as reference. To bring everyone up to page, I give a quick introduction of how this liftover is working. It is designed to remap the coordinates between assemblies of different assemblies of one species, but can also be applied across species for different genomes. And it makes use for that in chain alignment file that establishes the correspondence of the regions in the 2 genomes or the 2 assemblies. And with this, it can map the input coordinates, here are in purple, using this alignment file onto the output coordinates of the new genome. So that means for us specifically, we used the human ataxic data, we mapped this onto the bovine genome, and we're then comparing it to real data, allowing us to measure the correctness of these mappings giving us a quality measure of precision. And at the same time, we can also assess the amount of real data that has been recapitulated by this mapping, giving us a notion of sensitivity. If you keep in mind that both for human and for mouse we have multiple tissues, that means we can, for example, take human cortex data and map this onto the bovine genome and compare it to cow cortex data, giving us 87% precision, meaning that 87% of these regions do find an overlap with real data. And one can do this comparison all against all, and we find that there's generally a rather high precision, ranging from 37 up to 89%, but all that comes with a not very high sensitivity, ranging only from 4% up to 36% on these data sets. But of course, we are more interested in the tissue-specific mapping, so we're focusing here on the diagonal of this data matrix. And in order to compare different methods and different parameterizations, we just use these numbers. We even combine the precision and the sensitivity with a harmonic mean, which is called the F-score, and also take an average over the tissues in order to get a handleable amount of numbers that can be easier compared. So these three numbers here that you see in the last column, I now put here into this new table, together with some statistics of the method, where you can see that in the liftover process, 8% of the regions have basically been lost, so we're not being able to be mapped. That is partly because of no aligning sequence, but partly also because you need to keep in mind that these associations, they are not necessarily one-to-one, but one region can also map to multiple output regions, which are per default not included in liftover. But if we do so, then the number of regions lost slightly reduces, but it doesn't have a real impact though on our quality measures. What you though can see is that the number of bases that are being covered by these regions drastically increases on average, more than triples. And the reason for this is that liftover per default takes the boundaries of a region and maps them, and if within this region an insertion is present, like here in this example illustrated now, then such a region gets heavily inflated. PSL map, on the other hand, the other tool, is doing a different approach, namely a base-by-base mapping, and the output of a PSL map on this would mean that this region that I just was talking about would be split into two regions, but that would exclude the insertion. And this you can also appreciate in the change in the number of bases that is now in correspondence with the loss of the regions. And besides this, though, the quality measures of PSL map are similar to liftover due to the lower number of bases, slightly lower precision. PSL map also has a different parameterization option by taking in a different chain file. That's a technicality I'm happy to discuss with anyone interested offline, but it does have only subtle but rather consistent influence on this. What we don't now wonder is, I have mentioned that we are using here an alignment as a necessity for this process. So we are relying to some certain level of conservation. And we wondered if we introduce more conservation, for example, by restricting ourselves to constrained elements, as for example defined by GURB in the ensemble, in this ensemble alignment. What happens then? We are, of course, only able to map much less regions, like about a third less, and that obviously reduces the sensitivity as we have much less data. We can also recover less data. But of these regions that are being mapped, a much higher percentage consistently is overlapping with real experimental data. And we also can see that while now comparing liftover and PSL map on these conserved regions, both methods become in the output very, very similar to each other. But when I was looking at these tables, also not just at the overall average scores, but also throughout the tissues, there were some big variation present, which led us to the question of how does all of this is being influenced by the verification data, for which we then tried to use the newly generated BOFREC attack-seek data as reference, which on the overall output this F-score, you see there is no difference, but there are quite some differences throughout the different tissues in both precision and sensitivity that do overall average out. But the reason why they are there is because we do find a quite substantial difference in the attack-seek from BOFREC to the FANG attack-seek data. With this, I want to summarize that we have used the projection method to try to lift functional annotations from human as an example over to the cow with two different methods that have different side effects. A restriction to conserve the elements improves consistency, the precision, and of course does lower the sensitivity. But as a side note, it also alleviates the side effects of both methods. And in order to become free from the restriction of using alignments, machine learning is a promising approach. And all in all, as a summary, we have seen differences between the methods, between the different parametrizations, but all of this was very much in the range of the differences that we see arising from the experimental data, which I would conclude with that we should not overinterpret these differences, but rather aim for reducing the differences in the experimental data, which from an experimental point of view, of course, is challenging, but certainly straightforward, somewhat, or aimable from the perspective of the subsequent analysis steps. And with this, I would like to hand over to José introducing the topic. Thank you.
Any questions for Bjorn? Krista, am I?
Yeah, it's me again, Krista, but I really encourage all the other participants here on site to participate in the discussion. But one question which comes into my mind when I look at these data, you look more or less, you have the hypothesis that these areas are conserved. However, particularly in BOFREC, we wanted to go for extreme diversity. So we looked at different breeds, different ages, and so on. So what would be the key message? And in addition, you use data from humans where we don't know another species, we don't know the environment, we don't know anything. How much overlap do you expect at all? And what would be the interpretation of those overlapping regions? Because we are looking for the differences in functional annotation, making from one sequence all the different cell types, different environmental reactions.
Yes, right. From the from the evolutionary point of view, you cannot look at the differences. It is more about what is common in between the species, and only this common, so to say, this conserved information can potentially be mapped. But it is so. I would say, from the evolutionary point of view, one can use this conserved information as a starting point to start annotating novel species for which nothing is known so far.
So thank you, Bjorn. We already discussed that quite a lot of time. I think all the regions that are constrained evolutionary, you don't need BOFREC data to add new things. I think what is the richness of BOFREC data is what is unique to cattle. And so I don't think with a comparative analysis you will add something to these cattle evolutionary species-specific elements, which are really interesting because highly active and specific to cattle or to ruminant or
Yes, right. And what important was from the Bovrek point of view was, in order to establish such a method, one needs to, of course, have also data with which one can verify. And here, really, the Bovrek data plays, the experimentally created Bovrek data plays a big role.
How automated are the methods in terms of repeating when new references and new data becomes available? And do you think that some of the new references that are going to be coming online, particularly T to T, will make a big difference to the power of that analysis?
I cannot much comment on this. We only tested this on the 1.2 assembly. But as you do have for every new assembly also alignment files that are creating the correspondence, it basically just depends upon the quality of these. I don't think that the changes within one assembly are considering that or under the presumption that the quality of the assembly is good. I don't think that there are major influences that would outpace the differences between the species.
Hi, sorry, James Prendergast, University of Edinburgh. So by definition, liftover only can give you sites which are conserved between the two species, so the sequences have to be conserved. If you look at synteny rather than just sequence, can you identify peaks which exist despite the sequence having diverged? So you've still got a regulatory element where the underlying sequence has changed, but the regulatory element still exists.
I do not know how much additional information that would bring, because these chain files are already genome-wide alignments that partly do relate on syntenic blocks. So as long as between two alignment blocks the information does, so there are genome-wide alignments. So you have already information of synteny inside.
But so if you use the UCSC liftover file, only about 50% of bases can be lift over between human and cow. Mm-hmm. So there's about half of the genome you can't lift over based on these alignments.
So the recommended parameters here for cross-species lifting is 10% similarity, is what I also used.
Okay. Because, so you've got a very different parameter setting to the UCSC liftover file, is that right? Is that correct? You've used different parameters to the UCSC liftover file?
Now, you do have different parameter recommendations for doing a mapping between assemblies, where you are, I think, requiring 95% match. But if you go for mapping between different species, what I've seen the recommendations is min match parameter of 0.1, so 10% matching.
Okay, thanks.
Okay, so I think